Good morning, students. Today is another day filled with learning. Yesterday, we discussed about biotechnology, its definition, and how GMOs were created. So now, let us continue our discussion about biotechnology, particularly on its practical applications. Nowadays, several useful products are produced through biotechnology. Biotechnology has done a lot for biological research, medicine, agriculture, and even crime-solving procedures. So now, let us discuss some of the practical applications of biotechnology. One practical application of biotechnology is in forensic science. Do you know what forensic science is? It is the application of science or technology in the investigation and establishment of facts or evidence in a court of law. Blood, semen, hair, or small tissue samples may be found at a crime scene or on the clothes or other things owned by the victim or the assailant. Take note that sometimes these samples are available in small amounts. Since you need an ample amount of these samples, the DNA can be extracted and multiplied through PCR. DNA fingerprints are then made from these samples. This is a precise way of identifying the owner of the tissue or cell sample. No two persons have an identical DNA fingerprint except for identical twins. DNA fingerprinting can identify the guilty individual with a much higher degree of certainty. Your DNA fingerprint serves as your genetic ID that tells everything about you. Biotechnology is also applied in the production of human proteins using bacteria. Bacterial cells are producing proteins for us. How is it made possible? Are you familiar with diabetes? It is a condition characterized by too much glucose in the blood. One possible cause is the lack of insulin, a protein hormone that removes glucose from the blood. Did you know that insulin is the first human protein to be manufactured using recombinant DNA technology or genetic engineering? In this process, two DNA molecules are joined together and made to produce a protein. The protein is produced by inserting the gene that codes the insulin into the chromosome of a bacterium. The transgenic or transformed bacterium is then allowed to reproduce. As the bacterial cell undergoes successive cell divisions, the cell multiplies together with the gene that codes the insulin protein. In this way, large quantities of the protein are produced. Next is in the production of human proteins using livestock. There are disadvantages and difficulties encountered in the production of human proteins using bacteria. The transformed bacteria can produce complex human proteins, but the proteins are often not functional and active. What do you think is the disadvantage of this? It is impractical, of course. To solve this problem, scientists make use of livestock like sheep, cows, and pigs. Genus genes, a transgenic pig, were manipulated to produce the human protein C. The gene for protein C was inserted into the DNA of genus mammary gland cells. The glands produce milk with large quantities of functional and active protein C. The protein is now made commercially available for people who have digestive problems. Another one is in the detection and treatment of diseases. Medical scientists can now detect diseases using DNA technology such as PCR before the onset of symptoms or even before birth. Some diseases such as hemophilia and cystic fibrosis are caused by defective genes. These diseases can now be treated through gene therapy. The goal of gene therapy is to remove defective genes in the body and replace them with normal ones. These normal genes are introduced into the human body. Through this therapy, persons with genetic diseases will have a greater chance of living long and normal lives. Biotechnology has been also applied in plant breeding. Before, plant breeding was done by crossing the genes of two plants with desirable characteristics. This technique has been successful in producing improved varieties of crop plants, but it is time-consuming and complicated. Because of genetic engineering, these problems can now be avoided. Plants are inserted with supergenes to exhibit improved or better traits compared to their ordinary counterparts. These plants are therefore called transgenic plants. Transgenic plants may look like ordinary plants, but because they express DNA derived from other species, they have desirable traits that make them grow faster, last longer, and more resistant to infection. Biotechnology has also influenced animal breeding in the same way it has revolutionized plant breeding. Biotechnology has helped breeders produce better and superior breeds of animals as well as increase their productivity. The genes of these animals are manipulated to produce vaccines, antibodies, and growth hormones. In genetically engineered cows and pigs, there is a corresponding increase in usable meat supply per unit of food intake. Through biotechnology, super chickens are produced. These are chickens that grow faster with a higher quality of meat and can lay a greater number of large eggs. Current animal breeding programs employ the technique of eliminating inherited animal disorders through selective breeding. Alright, we are done discussing the different applications of biotechnology. And now, for your activity, search for the case story of the telltale fallow birdie. After reading this story, answer the following questions. Question number 1. What were the clues that led to the identification of the suspect? 2. Why do you think the discovery of the seed pods provided the most crucial clue? 3. What if a strand of hair was found in the truck bed instead of a seed pod? Do you think it will still lead to the identification of the suspect? 4. What if the strand of hair belonged to the victim? What other clues would you look for? 5. How did the investigator conclude that the suspect was indeed the murderer? 6. What do you think are the other uses of DNA fingerprinting? You can send your answer on my email flashed on the screen. That's all for today. Thank you and God bless. Goodbye!